but
Got it.
okay, hold on. Yes, okay, just got that. All right. I want to welcome everyone. Please be aware that I will be recording the presentation to post for those that could not make the presentation today. Welcome to everyone joining from all over the world. My name is Sabrina Merchant. I'm the SHINE Syndrome Foundation's Director of Science Programs, but I'm also a parent to a 10-year-old with SHINE syndrome. SHINE Syndrome Foundation is a nonprofit run by volunteers who are parents of children affected by SHINE syndrome. Our mission is to improve the quality of life for families and children impacted by SHINE syndrome through supporting collaborative research among international medical professionals, nurturing a family community, and raising worldwide awareness. The researchers who have worked with us and agreed to give the presentation for the Science of DLG4-SHINE webinar series today have kindly volunteered their time to help us as parents, caregivers, and patients understand the DLG4 gene variant and the tremendous amount of work and expertise required to go from recognizing a very rare disorder to understanding how it causes the disorder and identifying and developing therapeutics to effectively and safely treat the disorder. Today is the second webinar in the Science of DLG4-SHINE series. The format will be a 15 to 20-minute presentation by experts in the research who will present their current work in an understandable way for a patient and caregiver audience. First, I have some ground rules. Number one, the researchers have kindly volunteered for this, so please be kind back. The question and answer period should be kept in their scope of knowledge and may not be able to be answered to questions outside their scope of knowledge, such as insurance questions and possibly others. I have included everyone to come in on mute. After the presentations, put your questions in the chat and I'll ask some of the researchers. I have also gotten questions by email that I will also ask. At the end, a SHINE Syndrome Foundation representative will speak about other opportunities for involvement. With that, let's get started on the introductions to the presenters. John Hogenesch is a professor of pediatrics at Cincinnati Children's Hospital Medical Center and a prominent figure in the field of circadian biology. His research has led to key discoveries about how biological clocks regulate vital functions in the human body. Dr. John Hogenesch is renowned for his work in core circadian tools. His work focuses on circadian medicine, which seeks to improve health outcomes by aligning medical treatments with the body's natural rhythms. Dr. Hogenesch and his lab were recently featured in the New York Times Magazine article, The Quest by Circadian Medicine to Make the Most of Our Body Clocks, which highlights their groundbreaking research. The article explores how their work is shedding light on how internal clocks impact everything from sleep patterns to the timing of medications. Dr. Hogenesch's innovative research includes optimizing hospital environments and treating patients on schedules to better support their natural circadian rhythms. He has a strong focus on translating scientific discoveries into real-world solutions. His work offers hope for more personalized and effective treatments, particularly for patients dealing with sleep disorders such as DLG4. I'm also excited to introduce Jiffin Palouse, a research associate at Cincinnati Children's Medical Center, where he manages the lab of Dr. Hogenesch. Jiffin holds a PhD in biology from Texas A&M University and has a rich background in circadian biology, including published works on circadian rhythms, in gut bacteria, and the effects of aging on sleep. His expertise spans molecular biology, mouse behavior studies, and clinical projects, including sleep and circadian disorders. Jiffin plays a key role in coordinating NIH-funded research, training personnel, and supporting groundbreaking discoveries in circadian science. And finally, it is my pleasure to introduce Sharon Tamir, an aspiring physician scientist currently pursuing dual MD PhD degrees at the University of Cincinnati College of Medicine. Sharon is part of the Medical Scientist Training Program and is dedicated to researching neurological disorders. Her PhD thesis, conducted at Cincinnati Children's Hospital Medical Center in Dr. Hogenesch's lab, focuses on understanding sleep and circadian rhythm abnormalities in SHINE syndrome, specifically studying our DLG4 mutation. Sharon has a strong passion for building clinical work with cutting-edge research to improve patient outcomes. So we'll just go ahead and have Sharon go ahead and share your screen and we'll get started.
Yeah, thank you guys so much for having us here. Everyone can hear me okay? Great. All right. All right. So yeah, thank you so much for having us. We're really excited to talk to you guys today about what we've been working on in the lab, and particularly this novel mouse model of SHINE syndrome and what it has taught us and what it can teach us about SHINE syndrome on a biological level, on a behavioral level, and also what interests our lab, which is sleep. So, yep, my name is Sharon, and like Sabrina said, I'm an MD-PhD student in John Hogenesch's lab, and all of this work is stuff that we did in the lab. So first and foremost, we're going to talk about a bunch of stuff today, so here's the outline for what we're going to be chatting about. We'll start with a little bit of background on genetic causes of sleep and what are circadian rhythms, how are they even applying. We'll talk about the SHINE mouse model, how do we make the mouse model, what is it based on, and some of the gene and protein expression we see in the mouse. We'll talk about some behavioral characterization we did and what those teach us about the mouse and potentially about SHINE syndrome. We'll talk about the circadian and sleep characterization studies we did and what that shows us about sleep on the biological and genetic level. And then finally, we'll summarize everything together and talk about future directions for this mouse model and where we hope we can take this next and what we can learn next. So starting off, we'll talk about some background. So in the Hogenesch lab, we are particularly interested in studying behavioral genetics. So this is how genetic mutations actually go down the line to affect behavior. So here's a little schematic of what sort of like a normal gene to behavior pathway looks like. So you have your healthy, unaffected DNA that gets what we call translated into a healthy messenger RNA, which is then turned into a protein that has some sort of activity or function in the body that will then affect physiology and behavior. And when this process happens as expected, the physiology and behavior happens as expected. But then if you have a mutation, right, in the DNA, that affects the message that the DNA sends, which affects the protein that's created, which affects the protein's function, which is then going to affect the ability of the physiology and behavior to happen as expected. And in our case, the disease processes that we study and we're particularly interested in are ones that cause abnormalities in sleep or activity rest patterns. So let's talk super quick basics on circadian rhythms. So we live in a world that is rotating and has light part of the day and dark part of the day. And our physiology and our activity is actually really different in these two phases, right? So when it's light, we wake up, we're active, we're doing things. And when it's dark, usually we're sleeping or resting. And this cycle, right, from activity to rest, back to activity to rest, this happens over 24 hours, right? So this is a periodic 24-hour cycle. And you can imagine that your physiology is actually super different in these two different states, right? The kind of hormones and processes that you need to be taking place while you're awake are very different than the type of processes you want to be taking place while you're asleep. So, for example, here on the screen, I have a chart showing cortisol and melatonin levels. These are hormones that are important for regulating your awakeness and your sleepiness. So you can imagine that cortisol is something that we have a lot of in the beginning of the day, in the morning, because it helps us wake up, right? But it's very low at night when we're supposed to be feeling sleepy. And then melatonin's the opposite. It's really high when we're supposed to be feeling sleepy at nighttime, but it's really low in the morning when we need to be waking up. And these cycles of protein expression, of gene expression, these physiological cycles are really important for behavior, right? And you can imagine that genetic mutations in any of these processes will also affect our behavior and our ability to adequately respond to light and dark. And also at the bottom of each screen, I have a little explanation for what each of the words are if you have any questions. So now let's talk about functionally, what are circadian rhythms? How are they regulating that sort of behavior in light-dark cycles? So on the organ level, right, there are several things that tie in together to signal or cue to our body that it's a certain time of day. One that might be very obvious is light, right? Our exposure to light is going to then cue in to our body time of day. When we eat also factors in to cue to our body what time of day it is, and our activity, our physical movement, any exercise, anything like that also communicates to the body what time of day it is. And all of these signals come together to communicate to a specific region of the brain called the hypothalamus that we'll discuss more later. But the key point is that this region of the brain synthesizes all of that information and then communicates it to the rest of the body so that other physiological processes can occur at the appropriate time of day. So sleep-wake cycles or cognitive performance in the brain, these same processes signal to the blood to regulate blood pressure, which changes throughout the day. It signals to your metabolism, right? When should I be getting ready to metabolize food? It signals to your muscles, it signals to your kidneys. All over the body, we signal to these different organs. And then within the organs, there are actually cellular clocks. So every single cell, almost ubiquitously throughout the body, has its own mini molecular clock that receives these signals from the central clock in the brain and then synchronizes to that clock. So the way these molecular, smaller cellular scale programs work is it's sort of this genetic program that involves a core set of genes pictured here that are activated, create proteins that then inhibit the proteins that activate the gene expression, and then that stops protein creation, which stops the inhibition, which allows for the protein creation to happen again. And then this takes place over the course of 24 hours. So really quick, I'm actually going to hop out of here and show you a really quick video that sort of shows us a little bit more dynamically. So can everyone see the YouTube screen here? Excellent. So we have these genes that then express these proteins, right? And then these proteins are going to get made. Let's see if we can speed this up a little bit. Yeah, so the proteins get made, and then the proteins come back into where the genes are located, and they're going to affect gene expression. And this whole process takes about 24 hours, and in that way, our individual cells can keep time. Okay, let's come back to the slides. All right, so if you have any questions about any of that stuff, we can absolutely answer it at the end of the presentation. It's a little bit complicated, so no worries. Next, let's talk about the SHINE mouse model that our lab made. So in our lab, we were particularly interested in using a variant of SHINE syndrome that was in a patient that comes to our circadian and sleep clinic at Cincinnati Children's Hospital, because this patient exhibits all of the key symptoms of SHINE syndrome at a relatively severe level. So here on the screen now, you see the DLG4 gene sort of schematic, which encodes postsynaptic density protein 95. And the mutation that we're looking at is on what we call the C-terminal end, or near the C part of the protein, as you can see on the screen. And this is the variant. It's V692 transferred to tryptophan, or W, with a frame shift of 12. So what we do is we take this, this genetic variant that we want to make, and we used what's called the CRISPR-Cas9 system to insert that mutation into a mouse model. And more or less how this works is it's sort of like a molecular scissors that allows us to specifically target the healthy gene in a mouse and replace it with our mutant of interest. So we identify the mutation in the human gene on the mouse, and then we use these molecular scissors to do that specific targeting. This is a really long process. Just generating the first mouse takes like 6 months. And then in order to grow up the mice to a colony sufficiently large enough for us to start doing some scientific experiments and really getting into the sort of the mechanism of trying to understand these disease processes, that could take another 6 months or even up to a year. So yeah, so this is a long process. But some things that we've learned off the bat already, which we thought you guys would find really interesting, is that these mice do have a normal lifespan, and these mice have normal weight gain. And both of these properties are pretty important to show that there is some healthy aging going on as far as like physical, like purely physical responses in these mice. All right, so let's talk a little bit about the molecular characterization we've done on these mice so far. So here again, I've put on the screen this sort of schematic that we talked about, how we go from changes in the gene to changes in physiology and behavior. And one of the things we wanted to look at is to see, is the gene expression, are the messages that the gene sends, the messenger RNA, how is that expressed in the SHINE mice or the heterozygote mice that have one copy of the mutated gene and a copy of the healthy gene, like most SHINE patients do? How is that expressed in the mice? Is it what we expect? So here, now what we're seeing on the screen is on each row, you're seeing this is messenger RNA probe looking at healthy, unaffected DLG4, so unmutated DLG4, and then this is a probe looking for mutated DLG4. And each column is a different type of mouse. So in the first column, we have unaffected wild-type mice, so no mutations. In the second column, we have the heterozygote SHINE mice that have one mutant copy and one normal copy of the gene. And then in the third column, this is just a no sample control to make sure that if we do see any signal or any of these bands, it's not because we did something silly on our end. It's actually it actually is what we think it is. So what we're seeing here is that in the wild type unaffected mice, we're seeing expression of the healthy, normal, unmutated DLG4 messenger RNA. But we're not seeing any expression of the mutant. And this is good because the healthy, unaffected mice should not have any of the mutant copy. But interestingly, what we do see in the heterozygote or in the SHINE mice, we see some expression of the healthy RNA, which is expected because they still have a copy of the healthy RNA. But we're also seeing expression of the mutant RNA. And this is something that one of our collaborators, Dr. Toomer, who talked in the previous session, found in our patient. So this is interesting because normally the body has processes in order to make sure that RNA is not made from genes that are mutated. But this process is subverted both in our patient and in our mice. So this was interesting to us because it shows us that our mice on a molecular level behave similarly to our patient. All right, and let's talk about protein expression. So we know that DLG4 encodes this protein PSD95 that we can see here. And so here on the y-axis, we're looking at different proteins where This top region is going to show us PSD95 expression, and this bottom region is showing us this protein called GAPDH. This is what we call a loading control, which means that it verifies to us that we put the same amount of sample in this column as well as this column. So any changes we see in protein levels are not due to the amount of sample, but are actually due to the expression of that protein. So in the first column, again, we see this wild type or unaffected mouse. And here we see some clear signal of PSD95 expression. And when we look at the heterozygous or SHINE mouse that's expressing one mutant copy of the DLG4 gene, we see a marked decrease in PSD95. So this shows us that there is less PSD95 protein expressed in these mice, and this is also very similar to what we see in our patient. All right, next let's talk about some of the behavioral tests and assays that we did on the mice and what those tell us. So the first test that I'm going to talk to you about is called rotarod. And this stands for rotating rod. And it actually is this rotating rod that we put the mice on, and the mice sort of try to walk on it. And then depending on how good their motor coordination is, they'll be able to walk on it longer, or they might fall off. So I'm going to show you a really quick video of what this looks like. Okay, so here we go. So here you can see the mice, they're on the little rod, and then, the ones that aren't as good will fall off. So no harm to the mice. I just thought you might be interested in seeing how that looks. So when we put our wild type or unaffected mice and our heterozygote or SHINE mice on these rods, and we looked at their ability to stay on the rod while it rotates, we found that the heterozygote mice or the SHINE mice struggled with staying on the rod more than the wild type mice. So here what you're seeing is latency in seconds is just how long were they able to stay on the rod. And in this green bar, you see that wild types were able to stay on average longer than the blue bar, which is the SHINE mice. The next test we did is called a startle test. So this is a behavioral assay that measures sort of your reflexive startle response. So think about it, Halloween's coming up. If you're walking down the street and someone's got a spooky yard in their front yard or something, and they jump out at you and scream boo, you're probably going to have this like reflexive sort of startle response. Mice do the same thing. So let me show you a quick little video of what it looks like when we do the startle response on the mice. So here what you'll see is a mouse just hanging out, and when you see the little yellow X come up in the corner, that means the startle stimulus happened. Usually it's a loud sound, and you'll see the mouse do a little whoop, like a little startle response. So yep, there it was. And there it was. So these are really interesting because they are a reflex. This is something that we've characterized really well in the field and we really understand. And it tells us also about the neuronal communication that allows for this reflex to occur. And because DLG4 PSD95 is expressed at really high levels in neurons, any changes to this could indicate some changes to neuronal function, potentially. So the first test we did is a tactile startle reflex test, which instead of a sound is a little puff of air that goes on the mice. And when we did this test, we found the following. So first of all, Vmax is it's a measurement of the degree to which the mouse was startled, right? How strong was the startle effect? And we see that in the wild-type mice, or the unaffected mice, this effect was stronger than in the SHINE mice, or the heterozygote mice. So the heterozygote mice had a decreased startle response to tactile stimulus, right? When we looked at the startle response in the mice, when the startle stimulus was an acoustic or a sound stimulus, we found that the SHINE mice had a stronger response to the sound stimulus. Now, this is interesting because the neuronal pathways, that's what these little black lines are pretty different for these two types of sensory stimuli. So this could determine some atypical sensory processing in these mice. The last test that we did, the last behavioral test that we did, we've got some more tests coming, is what's called this Morris-Water maze. And this is a behavioral assay that measures learning and memory. And the way it works is we put the mice in a little swimming pool, and we teach them that there's a platform, a hidden platform in the swimming pool, and their ability to remember where that platform is and swim to it is a measure of their ability to have learned where the platform is and to remember where the platform is. So the particular measurement that we are interested in is path efficiency. And you could see that the path could be very inefficient for the mouse, right? Like in this solid line where the mouse kind of swims all over the place and then eventually gets to the platform, or it can be a much more efficient path, like in the dotted line. So when we put our unaffected wild-type mice and our SHINE mice in through these tests and we measure path efficiency, we see across the board that the unaffected wild-type mice, the SHINE mice do worse, have a lower path efficiency than the wild-type mice or the unaffected mice, suggesting that they have some challenges with learning and memory and some potential intellectual disability, neurological disorder type phenotype. All right, so now we'll transition over to some of the specialized tests that our lab does and is particularly interested in, looking at circadian and sleep characterization. So first, let's chat a little bit about some tests and studies that the lab did before I even joined, sort of even suggesting that DLG4 could be important in modulating or affecting circadian rhythms. So this first test showed that DLG4 is expressed rhythmically in this region called the suprachiasmatic nucleus of the hypothalamus. So earlier we talked about how the hypothalamus is this region that synthesizes information from light, from feeding schedules, from activity, and uses that information to determine time of day and then communicate that to the rest of the body. And this SCN, or suprachiasmatic nucleus, this is the particular region of the hypothalamus that is really crucial for doing that work. So this is a part of the brain that our lab is really, really interested in. And what you're seeing here on this particular graph is this is DLG4 expression here on the y-axis. So this is levels of that messenger RNA, that messenger that comes from the DNA. And on the x-axis, you're seeing what we're calling circadian time. So when it's dark is when it's typically dark. When it's light is when it's typically light. And you're seeing this happen over the course of about 3 days with samples being taken every approximately 2 hours. And when we take these samples and we look at the expression, we see that it's rhythmic. in the way that we expect something that is regulated in a circadian fashion. Another screen that was done in our lab was looking at what happens if you decrease that messenger expression of DLG4 in cells. Now, it's interesting to note that these cells are not neuronal cells and they're not brain cells. These cells are actually brain cancer cells. And the reason why we use them is because they have very strong circadian rhythms. So if there's any genetic manipulation or other sort of mutation or treatment that causes a change in circadian rhythms, these cells are really good at showing that. So first of all, it's interesting that DLG4 is expressed in these cells because historically, DLG4 is expected to mostly just be expressed in the brain. So this suggests that DLG4 might be having a role in other cells, maybe in circadian rhythms. And when we look here, this first graph is showing line traces of real-time imaging of circadian rhythms. And the way we do this is we attach a bioluminescence reporter to a gene that is very, very important in circadian rhythms. That is like the circadian rhythm gene. It's called BMAL1, and John actually discovered it, which is very, very cool. And we can see that in cells that are affected by this DLG4 siRNA, which this is the tool that we use to decrease DLG4 in cells, we see that this blue line looks pretty different from these pink and green lines. And when we look at the period or the cycle time of all three of these different treatments, we see that when we decrease the amount of DLG4 in the cells, it actually changes this period by almost 2 hours, which is a pretty significant difference. And this is compared to these controls. So really quick, this control here, sham siRNA, it means it's the same type of technology as the DLG4 siRNA, but this one isn't coding for anything. So it's not targeting any protein. It's just to make sure that our experimental process is working as expected. And this GFP positive, stands for green fluorescent protein. And this is to make sure that our bioluminescence is working as expected. So these two treatments are effectively unaffected. This one has decreased DLG4, and we see this change in period, which is really, really interesting. One of the first studies we like to do in our lab to understand these sleep-wake cycles, these circadian cycles, is wheel running. Mice love to run on wheels, and it's a little bit of an addictive process for them. So when they're awake, they like to hop on those wheels, and when they're asleep, they're not on the wheels. And in this way, we can sort of determine when they're active and when they're not. So this is a behavioral assay that we use to record locomotor activity. What you're looking at now, this is called a double plotted actogram. So actogram meaning acto like activity, gram like graph, something that is visual. And what you're seeing here in each row is actually 48 hours. So you're seeing 2 days side by side. And the reason we do that is because it helps us see trends better. And when you see this bright neon yellow, that means the lights are on. When you see this white space, that means lights are off. So we have a 12-hour light cycle and a 12-hour dark cycle. And it just continues again and again. These little black ticks, what you're seeing is every rotation of that wheel in the mouse's cage. So that means the mouse is going for it. And we know that mice are nocturnal, so we expect to see lots of activity in the dark cycle and almost no activity in the light cycle. So what you see here in this section, this is constant darkness. So what we'll do eventually is once we've got data on the mice in their light-dark cycles, we'll turn off the light and just see what does their innate circadian rhythm, unaffected by external cues from light, want to do. And this is really interesting because this can show us some changes that might be hidden when the lights are on. You'll also notice that this, instead of being a straight line, starts being an angled line. And this is because our innate circadian rhythms, unaffected by external cues, are actually longer than the ones that are affected by external cues. And those external cues are actually really important for keeping our circadian rhythms at 24 hours. So now let's compare the wild type to the heterozygote or the SHINE mice. First, we can see it looks like there's far fewer black text, right? It's maybe a little bit less active. And then there's also a little bit of this running into the light, which is interesting and different from the wild type. And when we actually run the statistics on these behaviors, we see that the average activity is decreased almost across the board in the light dark and in the dark dark conditions for both male and female mice compared to the wild type or the unaffected, except for male mice in constant darkness. The blue is male SHINE mice, and you see that they're actually more active on average than the unaffected wild type mice, which is really interesting and could suggest a circadian abnormality. The next study that we do in our lab is this, what we call is piezo sleep study. This is an assay that allows mice to sort of behave normally in their home cage, but we put a very, very sensitive, basically pressure pad on the bottom of the cage so that every time the mouse's chest hits the floor and therefore the pad, we can record that. And interestingly, sleeping mice have a very particular 4 Hertz breathing pattern, right? So you can see here that these measurements of their chest hitting the pad is very, very regular. But resting mice or awake mice that are not asleep have this very irregular pattern. And in this way, we can actually record these sort of processes in the mice to the best of our ability without having to do any surgeries, implant any EEG recording or anything like that. So we like to do this as a first step at trying to understand sleep in our mice. When we put the mice through these tests, we find that our SHINE mice, for an interesting reason, have a longer sleep out length during their resting phase. And we're not entirely sure what this might mean. It's possible that the SHINE mice require more time to go through their sleep cycle compared to the unaffected wild-type mice. It's possible that the SHINE mice are more difficult to awaken or less easily awakened compared to the unaffected wild-type mice. But either way, this is an interesting phenotype that we're interested in delving further, especially with some EEG studies that I'll talk about in a little bit in our future directions. All right, so let's summarize everything we've learned today. So here's a table talking about all of the things that I've talked about today, and also comparing it to some degree what we know about the SHINE patient that this mouse was based on. So we know that our SHINE patient has gene expression or messenger RNA expression of the mutant gene. And we saw the same thing in the mouse, right? Thank you again to Tumor Lab for bringing us the data on the patient. And when we look at protein expression of PSD95, the protein that is encoded by DLG4, we see decreased expression in both the patient and in the mouse. When we look at some of the behavioral characterization or behavioral tests we did in the mouse, we know that the mouse has some impaired motor coordination, and we know that our patient, to a degree, also expresses some of this. And when we look at sensory motor reflexes, we see that peripheral sensation, like the tactile stimulus, right, is decreased or damped in the mice, while a more central stimulus, like the sound, something that comes from the ears, seems to be increased or augmented. And we know that our patient has some atypical sensory processing as well. And when we look at learning and memory processes, we saw some impairment in the mice, and we know that our patient has some impairments as well. When we look at circadian and sleep phenotypes, in the mouse, we see some hypoactivity, and we know that our patient has some ambulatory challenges. And when we look at our very, initial characterization of sleep, we see this increased sleep out length during the inactive or the resting phase. But in our patient and in most SHINE patients, we know that usually the issues are actually difficulty initiating sleep and frequent awakenings during the night. So, bringing this all together, where do we want to go next with this mouse model? We're particularly interested in some future experiments that we have planned, particularly EEG studies. This is the golden standard for how to assess sleep and also how to assess epilepsy. And these are both, and we're interested in both of these in this mouse, particularly because our patient really suffers from epilepsy. And we have a fantastic partnership with the Danzer Lab at Cincinnati Children's Hospital, who are absolutely paving the way in studying epilepsy. So we're excited to work with them to study our mouse some more, actually in this upcoming month. We're also really interested in understanding how neural communication might be affected in these mice. So we saw on the sensory processing or the startle tests that it looks like there are some differences. And we're interested in figuring out, are these brain cells signaling to each other effectively? We're also really interested in looking at some circadian processes. We are a circadian lab, so we want to do some more recording of real-time expression of circadian genes in cells and in tissue, like that suprachiasmatic nucleus, that particular region that's really, really important for circadian processing and behavior, to see if there are any effects on those regions in these mice. And lastly, and potentially most importantly, once we have a really good characterization of the mouse and an understanding that it does recapitulate what we see in the patients to enough of a degree that we can do this, we're really interested in looking at potential interventions. So partnering with the expertise that comes from our circadian and sleep clinic at the Cincinnati Children's Hospital and trying out some medications that could really alleviate, sort of in a root cause way, some of the difficulties that our patients experience. There are also a bunch of unanswered questions that are arisen from this mouse model, right? This is a mouse model of one patient's variant, right? So how generalizable is this model? We don't really know until we have more patient variant models and we can actually see what are the similarities or differences from one variant mouse to another variant mouse. And that leads sort of into the next question, which what is the range of the allele strength, right? Are certain alleles, certain mutations or variants stronger than others? How or why? So with all of that, I'd like to say a huge thank you, first and foremost, to the SHINE Foundation and DLG4 Fund for inviting us to chat with you guys about this work we're really excited about, particularly to the patients and the families that inspire all of this work, and absolutely to the particular patient and family that inspires my work. Also, big thanks to the Hogenesch lab, to Dr. Weaver in the Division of Human Genetics, Dr. Tom Dye, who I believe is here today, and Rochelle Witt, who've helped me a lot with this work, the Transgenic Animal and Genome Editing Facility at Cincinnati Children's that helped us make the mouse, the Animal Behavioral Core that helped us study the mouse, and yeah, and to you guys for having us here today. So thank you so much. We're really excited to take some questions. I'm throwing on the screen our contact information if any of you guys would like to reach out to us individually. And yeah, we're happy to chat.
I wanted to thank Dr. Tamir and Dr. Hogenesch and Dr. Palouse for this. This was a really wonderful, wonderful lecture, and I thought you did a great job of taking something so technically difficult to understand and moving it into and describing it in a way that I think all of us parents can understand it. So thank you so much for that. And then I'm going to go ahead and ask the questions that I got by email. And then if everyone wants to go ahead and put their questions in the chat, I can go ahead and answer them or ask them of the researchers. Sorry, Dr. Hogenesch and Dr. Palouse, do you guys have additional words you want to say before I start with the questions? And I just want to make sure that you're able to come off of mute. Okay, I think you're still on mute. Let me unmute both of you.
No, that was an excellent presentation. And we've made a lot of progress over the last couple years. And we're excited about delving more deeply into sleep behavior for this existing model, and also determining if other SHINE patients have similar phenotypes. Because if they do, then anything that works for patient one might work for patient two and save a lot of trouble. So that's the real goal here, is to characterize these models. And we can obviously try interventions in mice that we cannot try in people ethically. So that's another opportunity.
Thank you. So I'll go ahead and start with the questions. Some of them I think you've actually answered, so maybe it's just a reiteration. So how do the cognitive tests in mice compare to humans?
Yeah, so this is a great question. It is challenging. There are very few tests that directly translate from one to the other. So EEGs, the recordings we get are, at least to a degree, somewhat similar in humans and in mice, but for example, like a startle test, we don't really do startle tests on people, right? But we do other reflex tests on people. So it's not studying the same reflex, but it's studying a reflex, and the ability to extrapolate from one to the other is somewhat limited, right? Things like the water maze test, right? We do have learning and memory tests, but they do look really different from what we do in the mice, partly because mice can't really communicate to us very directly. So these tests are very well validated in the field, but the ability that we can, or the degree to which we can extrapolate from them, is still somewhat limited. So we take it all with a grain of salt.
Yeah, there is another assay we'd like to run called a 5-choice assay, and this is about higher executive function. We just haven't had time to do those studies yet. But yeah, there's more we can do that's probably more relatable, like somebody giving you a noise, and then if you go to the right window, you get in this case the mice get Yoo-hoo, that chocolate drink. But maybe you have something else that's more motivating for you. It could be a Merlot or what have you.
Yeah, we were talking about the swim test just on Friday or Thursday maybe, and John and Sharon both grew up around water, so they love swimming. I did not. I don't like swimming, so if you were to throw me into that Morris water maze, I wouldn't do well at all, and it would have nothing to do with learning and memory. It would just be that I don't like to swim.
I'm in your boat, so I understand this. So this was a question asked by a parent. And we'll see if you've ever studied this in cyclic mood disorders, maybe not just DLG4, but other cyclic mood disorders. The parent is asking, is there any link with bipolar disorder? Their son's sleep pattern changes dramatically on a cyclic basis, coinciding with mood change.
So I can't speak to this for sure, but just from my knowledge of the literature, there are some sort of case studies demonstrating that DLG4 or PSD95 mutations might be associated with bipolar disorder. So yeah, this is not the first time hearing of it, but I don't know a lot more than that, unfortunately, at this time.
There are a lot of relationships between clock disruption and depression, if that's what the parent is asking about. There's an entire disorder called seasonal affective disorder, particularly in northern and very southern climes where the day length changes, and a lot of people don't do well when they're constantly sleep deprived by light. I happen to be one of them. I need to get blackout shades, but I've been lazy for the last 6 years apparently and haven't done it. But I have a really hard time sleeping much past 6:30, 6:45 when the sun rises, which is a real wasted opportunity on my part.
I got mine from Amazon, so that's an easy way. So the three, and this is the last question from email. How do you use the animal model to develop therapeutics to help our children? It's pretty general.
Well, yeah, John, do you want to speak some more to this?
Yes, so I'm a therapeutics guy. I was at Novartis Pharmaceuticals for 6 years, and I've been thinking about therapeutics, including therapeutics for rare disorders, pretty much since. So for over the last, embarrassingly, over 20 years, I've been sort of thinking about this. And what the mouse allows us to do is to try interventions, and those interventions can be behavioral, exercise, diet, but also, as the parent asked, we can also try therapies. And so one of the ones we're excited to try is there's been a lot of interest in therapeutic intervention in general in the CNS, and there are opportunities for us to try pretty much whatever in this mouse model without having to go through a huge rigmarole with the FDA and trying to get approval to do initial studies. Even out of one patient studies are not nothing. We're good at it at Cincinnati, but it's not nothing.
Right, and this gives us basically the opportunities to try things like gene therapy. Can we reintroduce higher levels of the healthy copy of DLG4, and can that alleviate symptoms? Or pharmacological interventions? Are there certain sleep medications or other types of medications that actually return on a physiological or molecular level the sort of function of neurons or whatever else is affected? And then we can actually try that, study that, show the results. So yes, absolutely.
We could do gene therapy pretty much next month. That's an excellent point, Sharon. Would restoring normal expression to the brain be sufficient to alleviate some of these behavioral phenotypes? And if so, at what stage of development would you need to reintroduce function? And these are critical questions for any human therapeutics, because we need to understand, do you need to get a diagnosis at less than 1 year of age, or less than 5 years of age, less than 20, or does age not matter? And at any time it could work.
Yeah, I think that's interesting. I'm also wondering how the age of the mice compared to age of humans. How do you like if you see a certain thing in the mouse at like 5 months of age, what is that in humans?
Yeah, so a 5-month-old is almost an adult, pretty much, a young adult. It's not like dog years, unfortunately, because that's more relatable. So a healthy mouse can live up probably about 2 years, even longer in the right circumstances. So you can sort of think about it that way. Their development goes faster. So we can basically, if they're 6 to 8 weeks old, we can start to begin to study them.
Okay.
And that's a tremendous advantage. And that's the reason why a lot of people like other invertebrate models like Drosophila. But unfortunately, Drosophila for this type of thing isn't really appropriate because their learning and memory activities are pretty limited.
Those are fruit flies, by the way.
Oh, thank you.
Yes. I actually just saw one. I think the boys left the pineapple out.
Well, you should take them and do some research on them. So I'm going to go ahead and start the chat questions because we're starting to run out of time. Laura Palmer says, "Thank you, Sharon. That was an excellent presentation," and I completely agree. And then we have another. And the next question is, can you confirm that SHINE kids appear to require more time to complete their full circadian cycle?
That looks likely.
Yeah, so the activity from the patient that we have shows a fairly normal 24-hour rhythm. It doesn't seem like their rest activity patterns are any different. And the good news is that we see the same thing in the mouse model, is that there's no change in period.
Thank you. What can the SHINE community do to help further your research? For example, would it be helpful for individuals to provide you with case genetic variant information and EEG analysis data or something else?
Yeah.
I mean, I think thinking about the model, the second mouse model, the third mouse model, things like that, how generalizable are these findings? Because if there is wide disparity in the patients, we can expect that the models will also have a fairly wide disparity. And so these are critical questions.
Yeah. And then as well, of course, having a better characterization on a population level. What do we see in the SHINE kids, and what is the range of variability in these kids would also be really, really helpful. So thank you for asking that question.
And Dr. Toomer says, "Great work, congratulations. I look forward to further successful results. And I think we will learn much more about sleep problems and bipolar disorder symptoms when the deep phenotyping study is accomplished. So please, whoever asked that question about bipolar, try to get involved in the deep phenotyping study with Dr. Toomer so more and more information can be gathered about that." And then another parent, Anna, said, "Thank you. That was an excellent presentation." And they had a similar question as a prior one. And then the final question is, there seems to be a wide dispersion of phenotypes across the DLG4 population, sometimes a wide phenotype dispersion across identical variants. So some of the patients have the same variant and different phenotypes. My daughter, for example, is a heavy deep sleeper, 11 to 12 hours per day, whereas other identical variants appear to have disruptive sleep patterns where kids are waking up every several hours, multiple times per night. Your thoughts on that?
Yeah, so there's obviously tons of genes are influencing sleep, and it's likely that it's genetic interactions between the DLG4 allele and all the other alleles that determine the amount of sleep your body needs and the timing of that sleep. So we can also mimic this with mouse genetics. We've created our SHINE model, our first SHINE model, on a particular genetic background. But now that we have it on that background, we can move it to other backgrounds that might be more appropriate for cognitive tests, or more appropriate for sleep, or more appropriate for physical tests. So it's similar. It's as similar as we can get within the boundaries of ethical conduct of research and those types of issues. It's pretty much state of the art.
Thank you for answering that. There's one more question on the chat, and then afterward, I kind of did want to ask you guys about the sleep lab at Cincinnati Children's Hospital. So sleep patterns and period cycles. Our daughter on a monthly basis will not sleep for 2 nights prior to her period cycle, so her menses starting. Do you have any thoughts on the tie in between hormones and circadian rhythms?
Okay, so none of us are clinicians, at least the three of us. So
Tom and Rochelle are here as well, though.
right. I don't know
Yeah.
if they want to
Oh,
jump in on that, but honestly, I mean,
okay.
this is one of those questions where it's like maybe yes, maybe no. It would take a lot more to understand what's
There
going on
totally
there.
are sex differences between males and females in both sleep and in regulation of the circadian system. For example, my wife likes it to be extremely warm in the bed, and I cannot sleep at those kind of temperatures. So I need to throw off that side of the blanket and try to get cool, or it's going to be a long night.
And I think that Dr. Dye and Dr. Witt are on mute. I did, you can unmute if you like, if.
Can you give me this mute button that lets me shut down Dr. Guy? That'd be great.
Yeah, I can show you how to do that on Zoom. It doesn't work in real life, so.
Oh, that's terrible. That's what I wanted.
Um, Okay, so I think that the last question, or not, it's not really a question, just wanted to see if you guys could speak about just sleep medicine in general, and that would certainly be a clinical question about the lab at Cincinnati Children's Hospital, and maybe what they do and what parents could potentially in the DLG4 community, could they be referred to such a place at their local children's hospital, and what kind of information is obtained in that way.
Rochelle, would you like to take that?
I can. I was gonna actually just because Dr. Dye sees a number of these patients here at Cincinnati Children's, also ask if he had anything additionally to say. But I would say that we're both really fortunate to take care of children with complex sleep disorders in our complex sleep disorders and circadian disorders clinic, where similarly to the deep phenotyping that was talked about earlier, we're able to do that, I think, on a level specifically considering issues of circadian disorders and sleep. And then we have a couple of more tools at our disposal to then hopefully arrive at a therapeutic plan that's individualized and successful for each child. But definitely open to, if there were more precise or specific questions about what our clinic is like or what the sleep lab itself is like, where we do polysomnography or those types of things, we're happy to answer those questions as well. I just didn't know if there was a specific aspect of the clinical care that you were interested in us speaking more in depth about.
Rochelle's being super modest, but she's a sleep boarded neurologist along with Dr. Dye, who's also a sleep boarded neurologist. We run probably the largest clinic in pediatrics in the world. We see about 4,500 patients for polysomnography. This is where you go into a lab and they hook your brain up with EEG, but you also have other channels of information like how much you're moving, what your body temperature is, your heart rate. Those type of things are all sort of integrated in an evaluation of sleep. And this is something where, I don't like to throw out too many accolades, but our sleep group is really world class and provides world class care to patients, and also we have a pretty okay research effort.
Thank you. Yeah, I don't think anyone has any specific questions. I think it was just this is not available at every children's hospital or every community hospital. So just hearing about it and knowing that it's there that could help our kids with significant sleep problems is really important. So thank you for that.
I'm going to provide a counterpoint, a real brief one. I did the polysomnography myself last year, and I would have called it a not sleep study because I went into that lab sat in that dark room with all these wires and goo strapped to my head, and I did not have the best night's sleep ever. Kids do a lot better. That's what Rochelle and Tom tell me.
But I would also just, if I could add, how helpful this conversation has been, how helpful this conversation and expanded conversation in terms of hearing about the nuances of the sleep disorders or the sleep issues that children with SHINE syndrome have. It's so very helpful to us as clinicians and as researchers, because one thing that I notice is, even though it's a very prevalent feature of the disorder, its characterization. Are we talking about sleep fragmentation? Are we talking about switching days and nights? Are we talking about all of that information is really helpful to us in terms of better understanding the nature of the sleep disruption, or if it's quite variable across all of the kids. So again, a healthy collaboration with you folks is definitely would be desired by us as clinicians, because that really does matter and help us for certain.
Yeah, thank you. There is definitely a variation in our kids, and I
Yes,
think,
yeah.
and we all have a difficulty even describing what those things are. So that was really helpful, incredibly helpful. And one of the parents asked, overall, what would the process look like to get started, to get referred to you? Is it a primary care physician referral, something else? And then I'd like to add on, do you do kind of initial telephone consultations outside of the state of Ohio?
So that's a great question. So yes, the answer to the first question is yes. In terms of pediatrician referral would be a way to get started to get seen by us in Ohio. And I know that there are options for telemedicine visits as well, even though some of our aspects, so we have not only on our team clinicians, MD clinicians, but also psychologists, behavioral psychologists, sleep behavioral psychologists, who are quite helpful, and their range might also be somewhat restricted. So we would say contact, and we could work it out from there in terms of referral, and work it out from there in terms of what aspects of the team might be able to support clinical care for sure. But there are definitely, we see patients from multiple states. It just gets into the insurance piece and those types of things, but
all
would
50
be
states
glad.
and like 65 countries worldwide we see patients from. It was 1.4 million patient visits last year. And
Wow.
we're now at 19,000 employees in Cincinnati. So we're like the third largest employer or something crazy like that.
Oh, that's amazing. Maybe we can get that information and give it to our community as to how their physician can go about starting their referral, or where they can contact to start the process.
Right. We even
We
operate
would definitely do that. Yeah.
in
Thank you.
the Dominican Republic, Brazil, other places, Pakistan.
Oh, that's amazing. We have a worldwide community, so I think that they would really appreciate that. Thank you so much for that. There's a couple of other congratulations in the chat. I think I'm gonna unmute Mara so that she can talk about opportunities for SHINE syndrome or any other questions they may have. And I'm gonna also unmute Tim. They're both our co-presidents.
Thank you. I had a quick follow-up to getting referred from your primary care physician or pediatrician. In my experience, this is the kind of symptom that is often underdiagnosed and undervalued in terms of the impact on health, on family health, on behavior, on everything. Are there keywords or key symptoms that you can express to some of your regular team for them to understand the need to refer to a specialist, rather than brush it off and say, give him melatonin, or try this or that? Because I think that's the experience of a lot of our patients.
Tom or Rochelle, do you want to take this one?
I also would again invite Dr. Daisari if he has some keywords, because I agree with you, unfortunately, that in terms of, how different either insurers or others who are responsible sometimes for the management of care, how seriously they take sleep disorders can be a challenge. I don't know if the mention of circadian, so that possibility with it being an issue of sleep timing or sleep length, those types of things, maybe those particular buzzwords may be helpful in kind of putting a name to what you believe the underlying deficit is. But I think that you raise an excellent point just in terms of how sleep and sleep disorders in kids are viewed. It can be quite varied. But I don't know if Dr. Dye has other thoughts about ways to have a successful referral experience.
Apparently, Dr. Dye can't find his phone. So.
Yeah, I think there's some problem with the unmuting. So let me.
Okay,
Let's
sorry.
try again.
But
Oh,
we
there
can
you go.
also
There you go.
go ahead.
Go ahead, Dr. Dye. We are still unable to hear you, Dr. Dye.
He's unmuted on our end, but I think he's
Okay.
muted on his own.
Can you hear me now?
There you are.
Yes.
Yes.
Oh, okay. All right. Thanks. For some reason, it thinks I'm driving. I think that's part of the problem. I'm just sitting in my basement. But no, we also have the opportunity, if you don't need a referral, we take self-referrals at this institution if there's concerns, and then it gets appropriately triaged to if sleep epilepsy is the main concern, or if it's more of a behavioral or a circadian aspect, and then we would triage appropriately. We also have the opportunity to do combined EEG with polysomnography, so we can look at the EEG tracing to see if epilepsy is affecting sleep or if sleep's affecting epilepsy.
Okay. Yeah, I think, yeah, I don't know why the muting people keep getting muted, but if you need to unmute, let me know. Yeah, I think if maybe we have a pretty good private Facebook group, so I can kind of hopefully give that information to our community. If you guys have any particular like patient-facing information, I can hand that out as well. Thank you.
Mm-hmm.
So I want to respect everyone's time. I have Mara and Tim on unmute, so they can talk about opportunities within SHINE syndrome. And of course, thank you so much to our researchers. And you're welcome to stay on or drop off. We want to. Your time is very, very valuable, especially on a Sunday. So thank you again. Thank you so much.
You're just kindly noting that the NFL just started.
There's
The
the
1
method
o'clock
to my
games just got their
madness.
kickoff. How's your fantasy team doing so far, Sabrina?
Oh,
Tim's
yeah.
is doing good. He's winning.
Yeah, I'm not great at this, so no, not well. I make my husband actually like pick all the things, but not great there. How about you?
2-0 so far. We'll see.
Congrats.
Thank you guys so much for having us. We really appreciate it. And
thank you very
we're
much.
excited to continue working with you.
Same.
Sorry, I just got unmuted. Thank you, everyone. And anything that you folks can think of at Team Cincinnati, I'll call you, that where the community can be of more assistance, please don't hesitate to reach out to us. We want to be as cooperative as we possibly can be. This is great work, research, and great work. Thank you.
Yeah,
thank you.
Appreciate the time.
I truly think what you've presented will give people tools to approach their primary physicians to say those right keywords and express that this is something that needs addressing, and that's extremely valuable and important. So we're extremely thankful for your work.
Thanks, folks.
Cool. Look forward to hearing from you.
All right.
Yeah,
Thanks.
thank you.
Ciao, ciao.
And then to all the patient families who are around, as Sabrina was saying, you're hearing, you know, what's helpful to research. You're hearing world-class researchers working hard, showing interest and being helpful in, you know, all the ways we need them to be, and we're extremely thankful. Participating in registries is extremely useful for this. We were talking about difference in phenotypes. The Simon Searchlight has a sleep portion. So if we access all of those, if we had 50 phenotypical descriptions, same with Dr. Toomer's deep phenotyping study, please have the doctor who knows your child best reach out to her. The better we describe it, the better this research can be validated across the board, and we can find out the bottom line of what's happening, which it would be, you know, the next step obviously is addressing it the right way at the root, and so finding a treatment. And I think we can all agree, if the child sleeps better, the parents sleep better, and everybody, life goes better. So this would be a crucial symptom to address, and we're seeing a lot of questions about it in the Facebook group. So I think it's one of those very important issues. And get involved in the foundation if you'd like to. You see the great work that is being done, and we're, you know, on the front end of reaching out to these people and supporting them, and it's really rewarding. And of course, the last, as always, unfortunately, any fundraising, any support you can do. If we can give grants to the Hogenesch lab to further their research across more variants, we're going to get better answers, which will give us better outcomes for our children, for our families. So it's really important that we further that work and partner in all of these ways with the researchers. Tim, anything to add?
No, thank you, Mara. That's perfect.
Right.
And thank you, Sabrina.
Yes, thank you for organizing. We're going to try to organize more webinars, and yeah, so hopefully this is helpful, and the community, I hope, appreciates the content.
Thank you. Yeah, I'm hoping to bring more DLG4 researchers coming soon within the next several months. So hope to see you back.
Great.
Thank you so much, guys.
All right. Tell
Great
everyone.
weekend, guys.
Bye, everyone.